Early biology education introduces genetics through discrete binary switches. A pea is either yellow or green, round or wrinkled. But most biological traits, like crop yield, body weight, or human disease susceptibility, do not fall into neat categories. They exist on a continuous spectrum. In the 1760s, botanist Joseph Krollreiter documented this by crossbreeding tobacco plants. He found the subsequent generations produced an unbroken, continuous spectrum of flower phenotypes, completely blurring the lines between the parent varieties. Predicting these continuous traits is a massive economic necessity. Optimizing milk production in dairy cattle, for example, requires measuring and selecting for an extreme quantitative trait over multiple generations. This spectrum is driven by what statisticians call the infinitesimal model. Rather than a single dominant or recessive gene, a trait is simultaneously governed by multiple independent genetic loci acting together. The second major factor is the continuous, randomizing noise of the environment. Every organism is shaped by physical fluctuations in temperature, nutrition, and sunlight. This brings us to the foundational axiom of quantitative genetics. The observable phenotypic value of an organism is strictly the sum of its genotypic value plus its environmental deviation. To predict evolution or engineer better crops, analyzing the whole organism is insufficient. We have to mathematically extract the pure genetic signal from the environmental noise. When we zoom out from a single organism to an entire population, we stop looking at individual values and start measuring variance. This solid block represents total phenotypic variance, the total observable variation of a given trait across a population. We can deconstruct this total variation into three distinct components: genotypic variance, environmental variance, and the interaction between the two. The environmental variance captures differences caused by external conditions, encompassing both permanent lifestyle effects and temporary fluctuations, like a short-term drought. The genotype by environmental interaction occurs when different genetic profiles respond unpredictably to changing external conditions. A strain of wheat that thrives in humid soil might underperform a competitor in dry soil. This graphic shows broad-sense heritability. It is calculated as the precise ratio of genotypic variance divided by total phenotypic variance. Because it is a ratio of a part to the whole, its value strictly exists between 0 and 1. A high heritability score does not mean a trait is biologically fixed. It simply means that within a specific population and environment, a large proportion of the observed differences are due to genetics. In a laboratory setting, the challenge is isolating that genetic variance from the constant noise of the environment. Geneticists solve this by crossing two highly inbred homozygous lines to create an F1 generation. Because both parents are inbred and homozygous, the resulting F1 offspring are genetically identical to one another. With zero genetic differences between individuals, any variation we observe in the F1 generation is 100% environmental. In a standard experimental dataset, if the F1 variance is 2.0, we have successfully isolated the environmental variance. When those F1 individuals reproduce, genetic segregation occurs. The resulting F2 generation now contains both genetic and environmental variation. We measure the F2 generation and find a total phenotypic variance of 7.0. By subtracting the known environmental variance of 2.0 from the total variance of 7.0, we isolate a genotypic variance of 5.0. Finally, dividing the genotypic variance by the total F2 variance gives us 5 divided by 7. The broad sense heritability of this trait is 0.71. By mathematically zeroing out genetic variance in a control group, we can precisely quantify the total genetic impact on any segregating population. Broad-sense heritability is useful for evaluating clones, but it fails in evolutionary prediction because it includes genetic effects that cannot be reliably passed down. Total genetic variance is actually composed of three underlying components: additive variance, dominance variance, and epistatic variance. Dominance and epistasis arise from the interaction of specific alleles, either at the same locus or across different genes, creating non-additive phenotypic deviations. During the chaotic process of meiosis, independent assortment and recombination physically break these specific, complex genetic combinations completely apart. Because those multi-gene pairings are scrambled every generation, dominance and epistatic effects are not transmitted intact from parents to offspring. Additive genetic variance is different. It measures the cumulative effect of individual alleles that act independently, allowing them to pass cleanly through the filter of meiosis. This gives us narrow sense heritability, which calculates the proportion of total variation strictly due to that transmissible, additive genetic variance. This establishes a strict mathematical rule. Broad sense heritability is always greater than or equal to narrow sense heritability. Narrow sense is the filtered metric required to predict evolutionary change. Isolating narrow sense heritability provides the mathematical mechanism to predict genetic gain across generations, a concept formalized by animal geneticist J.L. Lush. He developed the breeder's equation, which states that the response to selection is equal to the narrow sense heritability multiplied by the selection differential. Consider a standard experiment measuring bristle number in Drosophila. The mean bristle count for the base population is 35. This graph plots the population distribution. We select a subset of parents with higher bristle numbers, averaging 42. The selection differential is the gap between the base population and these selected parents, exactly 7. We let those parents breed. Their offspring have a mean bristle count of 37.8. The actual genetic shift from the original base population is our response to selection, 2.8. Rearranging the breeder's equation gives us a realized narrow sense heritability of 2.8 divided by 7, or 0.40. Exactly 40% of the variation in bristle number is driven by additive genetic variance. The trait is moderately responsive to selective breeding. The breeder's equation turns an abstract statistical metric into a functional tool, allowing researchers to direct and quantify evolutionary change over time. Classical biometry can accurately calculate narrow sense heritability, but locating the specific DNA sequence responsible for that variance is a separate challenge. This discrepancy is known as the missing heritability paradox. Modern genome-wide association studies map individual genetic variants, but these localized points rarely add up to the total heritability predicted by classical family data. The current resolution to this discrepancy is the omnigenic model. The model states that complex traits rely on massive, interconnected regulatory networks involving almost every gene expressed in a specific tissue, rather than a few isolated core genes. Thousands of peripheral loci across the genome exert microscopic additive effects. Cumulatively, these scattered signals account for the missing additive variance. This brings the math full circle to the mechanics of natural selection, governed by Fisher's fundamental theorem. Critical survival traits, like fecundity, consistently show low narrow sense heritability. Because directional natural selection relentlessly favors advantageous traits, it quickly drives those additive alleles to fixation, exhausting the available variants. Heritability is not a measure of individual genetic destiny. It is a precise, mathematically rigorous snapshot of a population's variance at a specific moment, revealing exactly how much evolutionary potential remains locked within its additive alleles.